Speak Student!
Molecular genetics. Polymerase chain reaction.
A la Shmoop. Okay, we're on next lesson, DNA technology with Dr. Ruth Tennant. Another subtopic. How does the polymerase chain reaction, or PCR, work?
Great. So polymerase chain reaction, or PCR, is a way of amplifying DNA, starting from a very small number of molecules. And it happens in 3 steps. So the first step is you heat up the DNA. It's called denaturation.
Amplifying pieces of DNA, like why would amplifying meaning making them bigger, louder?
More copies of the same thing.
More copies.
Yes, more
Okay,
copies,
sorry.
exactly.
Okay, keep going.
All right, so you've got this denaturation step, which basically you heat up the DNA, it separates the 2 strands. The next step is called annealing. So you lower the temperature, and what happens here is you have little DNA primers, which are similar to the RNA primers we talked about before during replication. These DNA primers sit on either end of the gene that you're interested in, and they basically get things started for DNA polymerase. So in the third step of the reaction, when you increase for the extension, the polymerase binds and it extends those primers and goes across. So basically what you're doing is you're starting off with a single molecule of DNA, and then after one step, you get 2 molecules of DNA. So it's a way of amplifying every time by a factor of 2.
Got it. Then what's TAC?
TAC, yeah. So you can imagine that most proteins don't like getting heated up to 95 degrees Celsius
Yeah,
over
don't blame
and over.
'em. Yeah.
And so it's a thermostable, it's called. So it's basically from a bacteria that lives in like heat vents, and so it has no problem being hot for long periods of time. It's a special polymerase.
Got it. Okay. So why do scientists need to make copies of DNA in the first place? Like what is the end game here in making all these copies?
Yeah, the basic reason is because it's really hard to work with single molecules of DNA. So for example, if you wanted to figure out the sequence of a piece of DNA from one of your cells, it's a lot harder to do that if you only had 1 copy versus if you could make multiple copies. So it helps with that. It also helps if you wanted to clone. You can't really clone very well with a single piece of DNA, but if you had multiple copies, then you could be much easier to do that.
Okay. And then we combine the multiple pieces back together, and that's what is called recombinant DNA.
What is recombinant DNA?
Recombinant DNA is actually, you take a single one of those copies and put it into a new backbone. So recombinant basically means you're taking DNA from 2 different sources and putting them into one place.
Give us an example, like walk us through like one slip of DNA that would be an obvious use case.
Sure. Yeah, so one really cool thing that scientists have done is for people who have diabetes, they need to get insulin, and they used to get it from pigs and from cadavers, which is not ideal for many reasons. And so you can actually take the human insulin gene that you made through, say, RT-PCR, and you insert it into a backbone, and then you put it into bacteria, and then the bacteria will make the insulin for you.
Wow,
You don't have to worry
cool.
about getting it from pigs or whatever.
Okay, that's it.
How does PCR work? What is TAC? Why do scientists need to make copies of DNA? What is recombinant DNA?